Now that we've talked a bit about staining, increasing contrast, and why that matters, let's now get on to the next step, which is microscopy. And before we talk about microscopy specifically, let's talk about the size of things and why that matters. So on this particular slide, what we see is on this horizontal axis, we've got the scale of things. And so from 0.1 nanometer all the way to 10 nanometers. And what I want you to see down here first, right, the asterisk matter, is that your eye can see about 200 micrometers, and that's about 0.2 millimeters, as we'll see. So to see bacteria, which are in this particular range right here, right, you can see that our eye cannot go down as far to see that. So we need some type of microscopy. And so you are using a compound light microscope, which gets down to about 0.2 millimeters, which is the same thing as 200 nanometers. And throughout this course, we also see images that are taken from scanning electron microscopes and transmission electron microscopes. Okay, and so definitely know that viruses are smaller than bacteria, which are smaller than human cells. In general, this is red blood cell. This is one of the smaller ones. And so let's talk a little bit about unit of measurement. So you've all covered this, right, in various classes. And so if we go from a millimeter to a if we go from a meter to a millimeter, and a millimeter to a micrometer, and a micrometer to a nanometer, each one of those steps is a factor of 1,000-fold difference. So a millimeter has 1,000 millimeters in it. That's what milli means. Okay, a millimeter has 1,000 micrometers in it. 1 micrometer has 1,000 nanometers in it. Okay, so when we look at this, then these are different ways of showing how each one of these units of measurement relates to a meter, being the kind of the standard metric that we're using there. So 1 millimeter is 0.001 of a meter, and that's the same thing as saying 1,000, right? Because 1, 2, 3. Same thing as saying 10 to the minus 3 or 1 to 1,000. Okay, so know these, know how they relate to each other. And so, for example, how many micrometers are in a millimeter? Well, you take 1 millimeter and divide it by a micrometer. And so when we look at that then, this is 1,000, so 1,000-fold difference. And that's the key thing that I want you to know, is that every step when we go from meter to millimeter to micrometer to nanometer, we're going down 1,000-fold. Okay, and that's 3 orders of magnitude. It's a big deal. Okay, so how many micrometers are in a meter? I will let you determine that. And this is also why it matters. If you're looking at a particular cell that is 0.6 micrometers long, you should know that that's also the same thing as 600 nanometers long or 0.006 millimeters. And so bottom line, know how to go back and forth between these and understand the 1,000-fold difference between meter, millimeter, micrometer, and nanometer. So let's get into the different components here. And so we are using visible light. And so visible light right here is what a compound microscope uses, compound light microscope. And what we see that when we decrease the wavelength, right, the 700 to 400, as we decrease the wavelength, we have increasing resolving power. And without getting into a lot of the specifics, that's an example of why electron microscopy has higher resolution, and we can see much smaller things with electron microscopy due to the wavelengths. So if we look at some basics about a bright field microscope, compound microscope just means it has a series of lenses, okay, for magnification. In this case, when you look at the magnification, and we look down here, you have an objective lens, which we'll talk about, and we have the ocular lens, and that is the eyepiece. And you always multiply these, okay? So if you have an eyepiece that gives you 10 times the magnification, and you have an objective, which is 40x, then you multiply those, okay? You don't add them, and so the final total magnification would be what? In this case, it'd be 400 times magnification, total magnification, which means that when you use your immersion lens, your oil immersion lens, which is 100 times, you end up with 1,000 times magnification, right? So that's why we can see something that is 1 micrometer long through the microscope, because essentially we're seeing it as if it was 1,000-fold larger than that, a millimeter. right? So, and this oil immersion lens, which I just mentioned, increases the resolution, and we'll talk about why that is. Okay, so here is a diagram. Know all of the asterisks, know which those are. You should be able to work your way around a microscope and call each component the correct thing, right? So it's a tool you're using, so understand which components are called which. Now, if we look at the microscope you're using, so it's pretty much the same as what this previous image is, but the reason I threw these in, these are from this video that you're watching for lab, okay, where it goes into all of this. But I just kind of want to point out that the condenser lens is up here and the iris diaphragm is down there. And the iris diaphragm is really your friend, and you'll find that when you change magnification, as I talk about in the lab video, using that iris diaphragm to change the light or the slider can make a huge difference in what you can see. If all of a sudden you change the magnification and you can't see anything, it's black when you were just looking at something, go for the iris diaphragm and open that up, let that light in, and it'll work much better. Okay, so what I'm showing here is also from that video, and I'm looking at a letter E in there. And when I look at it on the stage or on the slide, I see that it is the right orientation. But in the microscope, I see, as you can see on this screen here, that it is upside down and backwards. Okay, why is that? Well, if we look at how microscopes work, they rely on convex lenses. And so by bending the light, if this is the specimen and light is shining through the specimen and through this convex lens, then basically it bends, it then crosses over at the focal point, and then basically if your eye is right here, you'll see the enlarged image. And due to the bending of light and the way it crisscrossed, that flips the image. Okay, and so that's why you end up seeing that way, upside down and backwards, just like this is. However, as I said before, your compound microscope uses multiple lenses. Some are convex, some are not. So if we look at this image down here, this kind of gives you an idea where the condenser lens, right, goes through, and it goes through the specimen at this point. So we've just crossed it once, right? So now it's upside down and backwards. And then we have the objective lens, your 4x, your 40x, your 100x, whatever the case may be, which also crosses it again. So in that case, it should be the correct orientation. However, look at this image one more time, and we see that in this case, the eyepiece lens then also diffracts it, right? And it will then bend it the other way and then make it so that essentially if you go upside down and backwards, upside down and backwards, upside down and backwards 3 times through, you end up still upside down and backwards. And so, so when you're seeing it, that's what's going on in the microscope. That's the point here. Okay, so now this is what we're talking about right now. The whole purpose of this is why you use an oil immersion lens with immersion oil. Okay, and that is because light is bent to different degrees with different components. So if we take a quick look at this beaker with this metal rod in it, what we see is that when we look at it through air, it goes down to about there. And then when we're looking at it through the water, it's in a different orientation, right? It's like it's been cut in half. And that is showing you essentially that the water is bending the light. And so this is just a nice way to kind of conceptualize what we're about to talk about right now. And that is that different media, such as air, water, oil, glass, they have a refractive index. And so how much light will be bent when it goes through, right, depends on the refractive index. And so air and water are very different. Glass and air are very different. And so you will get bending of the light. Immersion oil and glass are almost identical. So you don't have that goofy bending of light. It just keeps going through, straight on through, and that is the crux of what we're about to talk about. So if we're looking at this then, okay, we know we don't want bending. And if what we have right here is we've got a slide, And this is showing a cover slip and a specimen. So let's go ahead and take a look at an image from that one of the microscopy videos. And what I'm showing you is how you will do this. You will have the slide on there, and you see that the oil immersion lens is almost touching the glass itself, okay? And what you don't see right there is that there is a column of immersion oil right there. And so what we see here is that the objective is actually in the immersion oil, okay? It's touching it. And so what that's doing is that's creating a continuous column from the slide and your sample into the immersion oil. So in the case of this class, this glass cover slip is not there. Right, we're not using glass cover slips for the slides that we make. Now, the prepared slides that are in your microscope kit or in your lab kit do have a cover slip. But for the ones you prepare, it will be on there. So don't get fixated on the cover slip being there or not. Just know that essentially we have a continuous index of refractive index going from the slide through the specimen, through the immersion oil, and into the objective. And so if you contrast that over here, well, first see how all of the light is going through and it's straight on. Here what we have is when it goes from the glass to the air, it bends, and so light starts going away from the objective, and so you don't have all of the image clearly going through, and what that means is that you have a fuzzy image when you're looking at something really, really small. Now, you don't use the oil when you're using your 4x, your 10x, or your 40x objective, because the wavelength of light and the optics are all good so that they work together and you can see things very clearly. It's just that when you use that oil immersion lens and we're looking at things really, really small, we need that oil for that continuous light because we need all of the image we can get. We need to funnel that image into the objective instead of light parts going all over the place. And so if we look at, so that was increasing resolution. And resolution is the ability to differentiate between two objects. That's one example. Contrast, as I talked about in another video, is the ability to see something compared to its background, right? And we all know what contrast is. You may change contrast in an image and if you're making it look better. So we all understand that we're going for a kind of a punchy, wow, I really see that now, whereas before it was kind of all blending in. And so the blending in is like up here, and that's down here. And so this is important for resolution as well. And this is an example of resolution. This is an image actually from this class. And so what we see here is, okay, well, what is this, right? It's blurry. Why is that the case? Well, this is really something that looks like this. So it's really a diplococcus, okay? But when you have poor resolution, remember resolution means the ability to differentiate between two objects. So if we look at this, can we differentiate these two? Well, if we looked at this, oh yes, this is one cell and this is another cell, it would be very clear. However, with decreasing resolution as we go across here, we are not differentiating between this coccus and this coccus. They're kind of blending together and it now looks like a bacillus. And so that's what we're looking at up here. This was really a coccus right there and a coccus right there, but this is before immersion oil was put on, and so it was just kind of fuzzy as opposed to being whoop, nice and crisp and clear. And so that's why staining and immersion oil matter, is so that you can have high contrast to see it and then have really good resolution to be able to distinguish between two bacterial cells. And this image is just an example of why that can functionally matter, right? If you're looking under the microscope and you say, oh look, it's a bacillus, and it's not, well, you just got it completely wrong. Okay, so we went over gram staining already. This is just a placeholder to remind you of that, and that is a very routine stain. It's the most common stain that microbiologists use. Okay. However, there are others, and this starts going back to cell structure and function. So Mycobacterium, for example, Mycobacterium tuberculosum, that causes tuberculosis, if you try to gram stain, it doesn't work. Mycobacteria have this waxy cell wall, and that prevents certain stains from penetrating in it. Okay, so if you want to see them under a microscope to increase the contrast, what do? Well, a staining process called Acidfast staining was developed. And so, for example, if we look at this image, what we're seeing is we have acid-fast rods. That's what it's called when you do the staining and you end up with red and rods, just because that's what we call them. So red rods mean that you probably have a mycobacterium if this came from sputum. And so what we're seeing here is these are Staphylococcus, the blue spheres, and this right here is Mycobacterium. And so acid-fast staining is for mycobacteria. That's the bottom line. And so that's why this is a useful differential stain, because it differentiates other bacteria, right? So other bacteria will not be red. So only if you have that unique cell wall that mycobacteria have, and a few others, okay, will hold on to that stain. So. And here's just a few examples. So we've already talked about endospores in another section, and so you can see that. So before the bacteria completely degrades and releases its spore, as we talked about in another section, they're inside the cell still, and that's why they're called endospore. And so this is a differential stain where spores are stained green. And so if you look at that and you say, oh, look, there's a red cell and there's green spores inside of it, you know that that bacterium can produce spores, and so that allows you to differentiate different types of bacteria. And then if we look at this capsule staining, what we're seeing is that the background is stained in a negative stain, and then what we have here is the bacterium is stained and the capsule is not stained. So the absence of staining then shows you essentially that this bacterium has a capsule. And so we talked about what capsules are good for, right? For not being dehydrated, for being able to evade the immune system, to be able to stick onto things. And so this allows you to differentiate bacteria that do and do not have a capsule. And then finally here, a flagella stain. This one is really tough, but you can certainly get good results. We used to do, well, in microbiology class I teach here on campus, we've done all of these stains. And so this right here just shows a certain type of Peritrichus flagella. And so if we think about the staining properties and looking at bacteria under the microscope, we can say something about them. Oh, this bacterium is a gram-positive bacillus that has endospores and a capsule. That starts telling you a lot about that bacterium. However, if you want to identify it, you need other tools. And so there's DNA-based tools, which we may talk about later. But what I want to talk about right now is a phenotypic assay. And so this right here is an image of something we use here at JU down in the lab. And so basically you inoculate your bacterium in every single one of these wells, and after it incubates, some wells will turn colors, other wells will not. And basically what this means is that each one of those wells that is colored means that that bacterium is testing positive for that test. So this is a plate that's about this big that has a whole bunch of individual tests in it, and combined the proper order of, okay, these are purple and these are not, allows you to identify the particular bacterium. Okay, and so for example, these right here are if you can use certain types of sugar. So if you can ferment the sugar that's in here, it will turn purple, and then that's a positive. And so this particular bacterium could not ferment the sugar that's in here or here or here or here or here, but it could ferment this one and it could ferment that one. And there's a whole bunch of other things. For example, there's antibiotics, there's acids, and so this allows you to also learn more properties about the bacteria. Oh, look, it can ferment sucrose. Okay, so knowing things like that can then make you say, oh, well, if it's in the mouth and it can ferment sucrose to make acids, well then, maybe it's Streptococcus mutans, right? So here's a couple of images. Experimental results rarely look as clear-cut as what the manufacturer shows you, but these are a couple of plates from some bacteria from my research students. And so this right here, this is Janelle's plate, and she was looking at Staphylococcus aureus. That's what this means right here. And so this is from a different bacterium. This is from an unknown bacterium, and that's why it's called H7-1. And if now we look at the circles, this is just showing you that Staphylococcus aureus can ferment the sugar, but this bacterium cannot. Okay, Staphylococcus aureus tests positive for these assays, and this one does not. And same thing down here. They test positive for these assays, and this does not. So this is just an example of how two different kinds of bacteria have different patterns on this BioLog Gen 3 plate. It's called a phenotypic assay. And that allows you to then run that pattern through a database and ideally identify that. So what we're talking about, we have a nice clean order of events here. We're talking about staining bacteria. Okay, we're talking about gram stain as well as capsule, endospore, a variety of stains which allow you to start identifying different types of bacteria. And then if you have a phenotypic assays, you can then properly identify them. And so that's what we're kind of looking at here, although microbiologists typically don't do this dichotomous key. I think it's still useful from a teaching and learning perspective to look at this. And so you did a gram stain and you look under the microscope and you said, oh, it's gram negative and it's rod. Okay, and I've been growing it in the lab, so I know it can live in oxygen. And I just did a test, and it fermented lactose. And so you keep going down here, and in this particular example, the yes, no allows this particular bacterium to be identified as Escherichia. So from that perspective then, this is why all of these are useful. So not only from our perspective from the class learning standpoint of learning cell structure and function, why it matters, because every one of these staining and phenotypic assays tells you something about what the bacteria can do, but also microbiologists use these tools, use these assays, use these staining techniques to identify bacteria.